Hello, everyone, and respective STEM Expo representatives. I am Leo Russell, and today I will be giving our research project for BT 101 Class 050. The effects of histone deacetylase inhibitor, VPA, on correcting defective gene expression in SMN2, a study on valproic acid's effect on SMA patient cells. So, what is SMA? Spinal muscular atrophy is a genetic disease that affects young infants from living normal functional lives. There are two types, type 1 SMA without treatment, and type 2 SMA may survive but have severe defects. What causes SMA is an absence of SMN1 gene, and then a nearly identical copy of this gene, SMN2, has a defect of skipping exon 7 during RNA splicing. For this experiment, we use a histone deacetylase inhibitor that can correct the splicing of SMN2 using this experimental design. First, we perform cell culture, transfect our materials, isolate the RNA and synthesize cDNA, then perform PCR techniques with primer sets, lastly using gel electrophoresis to fully determine the expression of SMN2. Valproic acid is the histone deacetylase inhibitor we use to express this histone of our SMN2 gene, specifically HDAC2I. Materials include antisense oligonucleotides to bind to our RNA to express our gene results, testing ASO-N1 lipofectamine and ASO control scramble, which does not bind, and DMSO control dilute. Within our culture, we use two different cell types. GM03813 cells from SMA type 2 patient and AG06814 healthy human patient cells, conducting this using a 6-well plate. Using transfection, we can safely transfer our own nucleic acid into the cell. Our lipid-based lipofectamine transports our ASO across the membrane. Here's what our plates consisted of. Isolating RNA can help differentiate treatment more effectively in correcting splicing and understanding HDAC inhibitors in the process. We use a lysate to break down our DNA cell wall. These buffers include RW1 to remove all biomolecules and RPE to remove salt traces. Then we quantify our results using spectrophotometry. We generated complementary DNA from mRNA using cDNA synthesis. Using reverse transcriptase to produce cDNA copies, we used PCR techniques using thermal cycling. Agro's gel electrophoresis uses a positive and negative charge to help us see the DNA fragments in levels of base pairs. To conclude, the gel results show VPA's effect on our GMO cells. The increase in base pairs in the SMN2 gene shows our splicing was corrected and our exon 7 was included. Here we have the results from our flow cytometry images of anti-N1 before and after, and here we can see our SMN2 protein indicated by our S3 fluorescent dye. Thank you so much, and much thanks to Dr. Singh for his immeasurable guidance. Thank you so much for everyone at STEM Expo, and here are our citations. And have a nice day.